So this is lab exercise 37, which covers two phyla in the kingdom Animalia. The phyla include the Platyhelminthes, which are the flatworms, and the Phylum Nematoda, which are commonly called the roundworms. This is our cladogram here, and in the prior lab, we covered the porifera and the cnidaria, and now we're moving on to some what we call protostomes, and the Platyhelminthes. They're located right here in this diagram. This diagram is from your textbook, and the Nematoda, which are located right there. So you can see this diagram is quite complicated. It used to look much simpler, but some new information has caused a rearrangement of the phylogeny here. We'll talk more about that in the lecture. So let's just go ahead and move on. Here, these are your tasks or your objectives, and or things to do. So you're going to be looking at characteristics of the flatworms, Platyhelminthes, and nematodes, and specifically how they relate to those primary processes or functions needed for survival of these animals. You're going to need to be able to recognize and describe the morphology, the basic structure of these two groups. And you need to also know the characteristics that these animals have that are in common with the more primitive porifera and the cnidaria, which was in the prior lab exercise. We're going to also look at what characteristics of these flatworms and roundworms that are more advanced than the primitive platyhelminths and nematodes. And you're going to look at examples of both of roundworms and flatworms from some major groups within there. So in other words, you should be able to recognize some representatives from each group. And you need to also be able to define and distinguish between animals that are acoelomate, pseudocoelomate, and coelomate. And when it comes to the two phyla here, what are they? Are they acoelomate, pseudocoelomate, or coelomates? Because that's a significant structural feature in these animals. So for the introduction, make sure you go through the background, read it carefully, and when you do, you should be able to answer these basic questions like what two phyla are being covered here. Well, they are the flatworms, which belong to the Platyhelminthes, and the roundworms, nematodes. Now, platy means flat, and helminth means worm. And nematode, I'm not sure exactly what the actual translation of that is, but those are the two phyla. And when we look at what additional complexities do these have over poriferans and cnidarians, first of all, these guys are going to have 3 germ layers, embryonic layers, so that's triploblastic. And that means that all the tissues arise from 3 layers. We already saw 2 earlier in diploblasts, like the cnidarians, and that's the endoderm. And then we had the ectoderm. The endoderm tends to line like the inside of the digestive cavity, and ectoderm will make structures like the epidermis. But now we're going to have a middle layer, mesoderm. And mesoderm is going to be responsible for making tissues like muscle tissue. Now, the kind of symmetry here, once you get to triploblastic, this is something to remember. All the animals that are triploblastic, which are going to represent all the animal phyla we cover from now on after cnidaria, they're all going to have bilateral symmetry. And with bilateral symmetry, this is something to remember. They're going to have a more centralized nervous system. And so they're going to have a collection of neurons and special sensory organs towards one end of the body. And that end is going to be referred to the front end or the anterior end. And so we're going to refer to this as sort of a cephalization. Now, not all of the animals we're looking at are going to have a definite head region, but the tendency to have more sensory organs towards one end or the anterior end, which is the front end of this bilateral symmetry, bilateral symmetric animals. So this is something significant. Triplobasts are going to have bilateral symmetry, and that means you can only cut them down the main axis in one way and along one plane, and that gives you a left and right side. So the flatworms include tapeworms, which you may have heard before, and liver flukes. And their characteristics is they're fairly solid. They don't have a hollow body cavity or a fluid-filled body cavity. They're not segmented. Of course, they're bilaterally symmetrical. They have a digestive cavity with one opening. And for the roundworms, they include a roundworm in the genus Ascaris, which is a parasite of the intestine of humans and pigs. And then there's pinworms, also parasite hookworms, and filaria. All of these are nematodes that are parasites, important parasites to humans and animals. It should be pointed out, though, that although we're going to focus on these important roundworms, that many are actually free-living and important in helping break down nutrients that are locked up within larger organic particles. So they do help cycle nutrients in soil, for example. So there's lots of nematodes, soil nematodes, for example. These guys have a body cavity, but it's called a pseudocoelom. So they're pseudocoelomate. We'll explain what that is in a bit. They don't have segments. We'll see segmentation in some groups later on. Bilaterally symmetrical. They do have a tubular, complete digestive system. And what's meant by complete is they have two openings. They have a mouth and an anus now. The flatworms did not. So many live in soil or in aquatic environments, and some are important parasites that I mentioned earlier. I should point out that although not mentioned up here, there are many free-living flatworms, but there are also some important parasites, animal parasites, and they include parasites of humans. So moving on here, this is going to be the taxa that you need to know for this group. Of course, you need to know that we're still working within the domain Eukarya, and as far in terms of phylogeny, they belong to a supergroup called Opisthorchis, which is unranked. This is not a official taxon, but it's just a group within the Eukarya that represents phylogeny. So all members in this group, animal group, would, if you go back far enough, share that common ancestor. This is the kingdom Animalia, just as it was in the prior lab. But we're going to be looking at two phyla, the Platyhelminthes and the Nematoda. And the names you need to know are black bolded, so all of those mentioned here. There are subphyla that I wrote down here, and only one you need to know. In the past, the lab manual would just cover these three classes, the class Turbellaria, and then two that belong to a subphylum Neodermata. These two are strictly parasites. So they're parasitic, whereas the Turbellaria are sometimes referred to as the free-living because they don't parasitize other organisms. So these are the free-living flatworms. And a representative genus that we typically study is the Dugesia, and it's commonly called the Planaria. So here we see Planaria, it's the genus Dugesia. For the Neodermata, they have some unique features that allow them to become parasites within animals and evade some of the host animals' protective mechanisms and the host's immune system. And the genera you need to know is Opisthorchis, and its synonym for that is Clonorchus. So if you ever see in laboratory setting a slide labeled Clonorchus, you're talking about Opisthorchis. And then there's Fasciola and Schistosoma, and those are in the class Trematoda. You need to know that. You need to know that they're in the subphylum Neodermata. Don't worry about knowing the subphylum for the free-living flatworms. The trematodes are commonly called flukes. Okay, and the other class, Cestoda, these are it's the group that tapeworms belong to. And tapeworms are intestinal parasites. And one genus you need to know is Taenia. For that, it's an important parasite for many animals, including humans. There are many other genera. There's one labeled here, which is a very large human tapeworm parasite, tapeworm of humans. And then when we look at Nematoda, which we'll see in the second portion description of the lab, there's going to be genera Ascaris, Trichinella, Necator, and Enterobius are going to be important ones. Filaria we're going to talk about here, but in terms of lab exam, you're not going to need to know those. So just to add a little enrichment here, all of these are important parasites. Like I said, we're going to focus on nematodes that are important parasites, but there are plenty of free-living worms species that are just important, a part of cycling nutrients in ecosystems. So looking at flatworms of the phylum Platyhelminthes, they are acoelomates, and A means without, and if I were to just take a root out of there, sil means cavity. Okay? And so we saw that with Spongicell, right? So we're referring to whether or not these animals have a body cavity, and that means that there is going to be a fluid-filled space between the organs. And a body cavity like that is important because it allows for organs to move more freely and to expand. For example, if you were to eat a Whataburger, your stomach would get bigger and expand within your body. But if you're an acoelomate, that expansion is not as allowable because there is no cavity. All of the tissues are basically just jammed together, solid mass of tissues. Okay? So they're acoelomates, whereas the roundworms or the nematodes, the phylum Nematoda, those are pseudocoelomates, and they have a body cavity, but it translates to a false body cavity, pseudo, because there's a very specific definition for what makes a body cavity. And so we'll look at what that actually is here. So in the diagram to the right, we have acoelomates for an acoelome, which means no body cavity, and then we have here a pseudo coelome, and then we have a coelomic body cavity here. Here they've taken a section through a flatworm. You can see the head of the flatworm right here with two eye spots. Sometimes they call those ocelli. It's a pretty simple animal here. If we were to section through it, all of the layers, the ectoderm, which is the outer epidermis, the endoderm, which endoderm is always going to help line the digestive cavity there, and then the mesoderm, which is in the middle. All three of those tissues, you can see they're just packed within the body. There's no cavity. There's no space in there. Yes, there is a cavity inside the digestive organ there within the gastrovascular cavity, but there's no space between any organs. There'll be organs packed in here, as we'll see later, but there's no fluid-filled spaces. Whereas if we were to take a section through a roundworm here, you can see all three tissues, major tissues that will form the organs within the animal. You have your ectoderm, which is the epidermis. On the inside, you're going to have your digestive system there, and it's just endoderm. There's no other layers on the outside or the inside. Again, the inside of your digestive cavity or digestive tract is always going to be lined with the endoderm or gastroderm, sometimes they call it. And then here's your mesoderm. And what looks like empty right there where I'm drawing that dot, that's your body cavity. It's filled with fluid, and it's fluid-filled space between organs, say organs of the body wall and the intestine there of this roundworm. And that's your pseudocoelome right there. So you can see there they have it labeled as a pseudocele or pseudocoelome. And the reason it's false is because a real body cavity has a very specific definition, and that means that this fluid-filled space or that cavity needs to be completely surrounded or bounded by mesoderm-derived tissues. And you can see that on this side right here, we have the outside of the body cavity, we have the inside of the body cavity covering some major organ in there, like an intestine. There's on this side here of this cavity, there's no mesoderm there. If we compare that to this annelid, which is a segmented worm like an earthworm, when we took a section through it and looked at it here, you can see that it's coelome right in there. It has all three tissues, ectoderm, endoderm, mesoderm. But this body cavity that's there, you can see the endoderm here. This might be like its intestine. And there's the digestive tract in there where digestion occurs. You can see that both this side and the outside of the cavity, the inside and the outside of the cavity, are bounded by, there's boundaried or enclosed by mesoderm. Okay, so the definition of a coelom means completely enclosed by mesoderm. And we can see that the pseudocoelom does not have that situation. Then acoelom doesn't even have a cavity, right? So in this lab here, with the flatworms, we're studying acoelomates, and then with the roundworms, pseudocoelomates. And later on, we'll look at coelomates. Okay? So looking at the phylum Platyhelminthes, we see a nice diagram of a common free-living flatworm, which is Dugesia. So what are important characteristics that define Platyhelminthes? Well, they're bilaterally symmetrical. They're acoelomates. We already went over a lot of the other characteristics for this group. So they have no body cavity. They're triploblastic. They have bilateral symmetry. Many are free-living, some are parasitic, and we can go on and list all of the different characteristics that we were mentioning a while ago. So just make sure you take some time to answer that question for yourself. Go back and look in there. Even go back to this chart that's right here, and you can see you want to be able to distinguish what are key characteristics, right? No body cavity. There's another one that I just forgot to mention here. Their digestive tract system is not complete, it's incomplete. And that just means they don't have a mouth and an anus. So they're more like the cnidarians. Incomplete digestive system. So they only have a mouth. No anus. So that would have something similar. We give it a similar name, a gastrovascular cavity here. And here would be the mouth opening going into the digestive system for this. By the way, what we see here, and you should come back to this to help you answer some questions later on. They've taken a section through a flatworm like this planarian here and gone through there, and you can see it has labeled the epidermis, which is ectoderm, the mesoderm here in the middle. This right here is just space because that space is right there with this tubular extension called the pharynx. And then you get into that muscular tube right there, and that's what you see here. You're still going to have outer tissue right there, just like there's outer tissue here. That would be ectoderm or mesoderm. Ectoderm or the epidermis. And then in here, this is inside the digestive system. There's the cavity inside. So that would be endoderm. And then your muscle tissues in here would be mesoderm. You would come back to this diagram to help answer a question. You have to look at cross-section through flatworm. So the first class is Turbellaria. And what are some key characteristics of the Turbellaria? Well, there are some marine ones, and this one here is a freshwater one, which is the Planaria or Dugesia. They do have, they're like all the group, they're acoelomates, but they do have a more complex setup than the cnidarians. They have organ systems. And so when I found this diagram that's right here to show some of their different organ systems, they have a reproductive system, and their reproductive system, they have both male and female, so they are hermaphrodites. So hermaphrodite is kind of like monoecious, which they have both male and female parts. Now, a real goal would be to really promote genetic diversity through sexual reproduction, is to find a mate and exchange sperm with another individual. In a worst case scenario, I suppose these hermaphrodites, they couldn't find a mate. They could, I suppose, potentially self-fertilize, and some reproduction is better than none overall. They are capable of asexual reproduction if you were to cut them. I don't know if they actually do this on purpose or not, but if you were to split them down the middle, each side would produce an entire new individual. Here you can see the digestive system there at the top in green, which is it's a gastrovascular cavity with a lot of branching. A lot of branching is good for more surface area, more absorption. You can see the nervous system, and you have some sensory eye spots up here. These auricles out here are going to have special chemoreceptors for us chemoreceptionists, smell and taste. And you can see you have a ladder-like network of nerves going down the body, so on both sides and then connected from one to another. And then you have a high concentration of nerve cells or ganglia up there towards the front. So this would be what I was talking about earlier. You have a higher concentration of sense organs and a nervous tissue towards the front or anterior end. And I called that a little bit a while ago cephalization, right? Now, if you were to section through the body of this animal here, this is a diagram of some of the things you might run into. Here's the gut or the gastrovascular cavity. You have mesoderm, ectoderm. You have cilia, which would help them move along surfaces here. Those are the two nerve cords that are on the belly side of it. And then you can see the section through at least some of the male and female organs in there. So, looking at the genus Dugesia, you need to know that one. Again, it's commonly called the Planaria. So if you ever get asked about Planaria, they are talking about the genus Dugesia and asking what are some of the characters of Dugesia. And well, it's one of the things. It's a free living. It's fresh water. It has all the characteristics we were mentioning earlier for flatworms. And it asks, how does Dugesia ingest and digest food? And so we can get a handle on that by doing procedure 37.1. And there you're going to be looking at a living Dugesia and watch it go about its business of moving around and acquiring food. So I found a video that's right here. We'll let it play and see if we can observe some of the movements of this animal and watch it feed.
It is in flatworms like Planaria that we first see bilateral symmetry and cephalization, the localization of the sense organs in the head region. A pair of ocelli, light-sensitive eye spots, are located toward the center of the head. Lobes extending from either side of the head contain chemosensory cells. Planaria feeds using the protrusible muscular pharynx. The chemosensory cells on the lobes are used to locate the food. Through rhythmic contraction of the muscular pharynx, Planaria pumps the food into the gastrovascular cavity with a siphon-like action.
So pretty neat video and watching the live Planaria. So that's the best thing to not being in the laboratory and actually having a specimen to watch, which would be pretty cool actually if we could do that right now. So continuing with that procedure, after observing the live Planaria or individual Dugesia, you're supposed to look at a whole mount that's been stained so that you can see the gastrovascular cavity, which you can see on the right. That's labeled as whole mount right here. To take a look at it. So here, those lobes they were talking about with the chemoreceptors are called auricles, which is kind of what we use to say external ear, because it looks like a little ear-like extension. And then you have your ocelli, which we can also call eye spots. Here's the pharynx. This would be the opening to the pharynx, the mouth there. And there's a little bit of space between the pharynx and the body there that it's not part of the body. There should be nothing there. And then the back of the animal, this is posterior, this is the anterior end where the head is. You can see how that gastrovascular cavity has been stained or injected so that you can see it. You can see it's very, very branched. And then the posterior end has two branches there. So you can see all of that structure. This would be your gastrovascular cavity. And if we were to section through the front right here, that would be this picture right here. Now, this is not in your lab manual, but you can see this is the anterior branch of the gastrovascular cavity, And then you'll have little sub-branchings that you see out here, or diverticula they call them, which are pockets within your digestive system. Then if we took a section right through the pharynx, that would be like the picture I said you should go back to help you answer some questions there. So you can see the pharynx here in the middle. There's that empty space there between the tubular pharynx and the rest of the body there. So there's actually no tissue right here. That's that space I was highlighting right up in there. And then you can see the two branches of the gastrovascular cavity right here and right here. And then towards the posterior end here, you have the two posterior trunks of the gastrovascular cavity. You can see that right there and right there. Okay, so I always recommend that you sketch and label your observations. In this case, you can do these sketches from these images that you see online. And let's move on. There's plenty of questions to answer about your observations of Dugesia, and ask what features distinguish the head from the tail, so you can think about that and answer it for one eye spots, right? Now, what are the differences between the simple eyes, or ocelli they call them? Ocellus is a simple an eye-like structure, ocelli plural. How's that different from animals that have more complex eyes? How does the head move different from the tail? So we would go back, watch that video, and just give a basic description. Now, did it look like the planaria was moving randomly or in a specific direction? And your answer there would be a specific direction, because once you have bilateral symmetry, these animals tend to move forward, and they use their sensory organs to detect if there's food or things they need or danger, to back out of that area or get away from danger. So these bilateral symmetry animals lead with their sensory organs into their environment. So it's very direct movement. And that answers that question E about that. How are they adapted for that? Well, they have those sensory, or the eye spots and the auricles, or those little lobe-like structures there on the anterior, and then have those chemoreceptors. We didn't get to touch a planaria with a probe in this video anyway, so we'll be able to answer that question. And we are also not, part of the procedure asks you to look at how they respond to flashing light on them. And then where's their feeding tube located? The feeding tube is that pharynx. It's on the bottom. Are they bilaterally symmetrical? The gastrovascular cavity of Dugesia, it's highly branched. And what is that for? That divisions within there, that increases surface area, better absorption, right? And then you go back to those basic questions about how do these animals feed, reproduce, or respond to the environment. And this one is about reproducing and then being monoecious, because they're hermaphrodites, right? So monoecious is another name for having both male and female sex organs. So planarians do have a head, and in biological terms, this is referred to as cephalization, right? And then you want to ask, well, how does that help with a planaria to be able to feed, reproduce, and respond to the environment? Now, the planaria doesn't have a specialized respiratory system, so that it can exchange gases with its environment. They're going to need oxygen and give off waste of carbon dioxide, like any animal would. And how would this be accomplished? Well, the planaria is pretty flat, so here this animal can exchange gas with its ectoderm and even its endoderm within the digestive tract there. Can just exchange, and since it's very flat, it's easy to pass needed stuff to cells that are nearby. Likewise, it's easier to get stuff out. So being flat is one way that they can get around not having a respiratory system. So now we're going to move on to the subphylum Neodermata, and we now get into a group with two classes that are parasitic. The class Trematodes, these were the ones I said were the flukes. We were looking at Turbellaria earlier. And so what are some characteristics of the trematodes? Well, basically they're all parasitic, okay? And they all live within other animals. And so they have all the basic characteristics we saw for Platyhelminthes, but this is a big one here, and they're parasitic. And many of them are going to have some complex life cycles involving multiple hosts. So they'll go on one part of the life cycle in one host and then move on to another host. We kind of saw that in protists when we were looking at Plasmodium, the one that causes malaria, where you had both human and a mosquito as part of the life cycle. But here's a big difference here. These flatworms don't actually have an epidermis, they have an epicuticle. So instead of having an outer layer of cells for protection, their mesoderm is going to produce the epicuticle. And this epicuticle is a protective coating. And this coating is made of so it's sort of a protective covering. And it's made of proteins and lipids. And these proteins and lipids, they're going to help keep these parasites, because they're living inside the body, they're going to help protect. They protect the parasitic worm, because these protein and lipids are resistant to digestive enzymes. So if this parasite was in, let's say, an intestine where there was digestive enzymes, it's resistant to that. But also, we have specialized immune cells that throw these explosive packets of digestive enzyme at the outer body of these larger parasites in an attempt to kill the parasite and break it down and digest it down. So this helps them. These are immune cells. This helps them evade those immune protective mechanisms. So it helps these parasites evade the immune system of the host. So they can live in there without being affected so much by the host's protective mechanisms. So here's a flatworm, a trematode of the genus Opisthorchis, and you should know this one. And this one, it's a liver fluke, and so that gives you some idea that it's either associated with liver or the digestive system within the host. Now, opus, this is a word power here. Let's learn this one. The root for this, opus, means toward the back. And orchis, I believe, is derived from Greek language, and it means testes. And testes is where sperm are produced. So that'll give you a clue here. This is, what does Opisthorchis mean? You can answer that question. It says, compare with the morphology of this trematode. So we come and we look over here. This is the head end right here. This is the anterior end where the mouth is. And you can see we have this gastrovascular cavity. It branches into two main branches, and it's hermaphroditic. So you have a male and female. You got ovaries, but look where the testes are. The testes are towards the back. They're posterior. So literal translation here, Opisthorchis, and you can see these little low branch-like testes back here where sperm are produced there. So this Opisthorchis, also called Clonorchis, I mentioned that earlier. And this is what it would look like under the microscope. So again, make some sketches and learn the name and associate it with this image. In case I ask you what is the genus. Now, Opisthorchis sinensis, or the Oriental liver fluke. The life cycles here, and I mentioned earlier, they have complex life cycles. I think I have a link right here. If you download, you can click on it, and it may take you to the Center for Disease Control website where you can see the life cycle. Now, I'm not going to test you on specific names of the different stages of the life cycle, but you should understand what is meant by saying definitive host and intermediate host. The definitive host also may be referred to as the primary host, and then secondary hosts. But I'm going to go with definitive and intermediate hosts here. And the difference here is the definitive host is the one where you find the mature parasite, and that means that it is got mature testes and ovaries and producing eggs through sexual reproduction. So this is the sexually reproducing stage. And you can see that in this case, the definitive host in this life cycle is the human. Okay. And so here's how the life cycle goes. The adult fluke is living within the body of the host or the human, associated with the liver, and these parasites produce tons of eggs. Like, that's one thing to know, because that's part of being adapted to being a parasite. It's going to be hard for your host to pick you up, so produce a lot of eggs, and that increases the chances of another host picking you up. So many eggs may never ever get picked up by a host and just remain out there in the environment until they expire or whatever. So your adult flukes living in there would produce some eggs, and then the eggs make it into the digestive system within the digestive canal, and eventually exit out the anus of the host. And so these eggs, which are going to contain a stage in the life cycle called the miracidium, and these eggs are going to be picked up by snails, aquatic snails that are in there. They're going to be eaten, and the egg-like structures with the miracidium are going to hatch within there, and they're going to go through some development and differentiation to form sporocysts. And the sporocysts are going to contain many iridia, and these iridia are going to contain within them many of these cercaria, which break free of the snail. So they exit the snail either through their body walls or through their anus, and they escape, and they're swimming. They have a structure back here that allows them to swim, and they'll swim and burrow into the body of a fish. So they'll literally burrow right through the skin, and in there they're going to encyst as a metacercarial cyst within the flesh, and then a human goes and eats the fish and then picks up the parasite, and the life cycle is completed. So you can see the life cycle is complex. That would make this human the definitive host, and the snail and the fish intermediate host. So you should be able to recognize the definitive and intermediate hosts for this exam. I'm not going to be testing you on the names of the specific stages here. Okay, you do need to know that the name, though. This is Opisthorchis. Okay, this is the Oriental liver fluke, Opisthorchis, and I just expect you to know the genus. You don't have to know the full scientific name. Since we already wrote Opisthorchis, that's when we write it again, we can abbreviate the genus, because we're still talking about Opisthorchis, the specific epithet is sinensis. So some questions to answer about Opisthorchis, asking you to compare the digestive cavity or gastrovascular cavity of Opisthorchis with Dugesia. And then they also ask you to look at the position of the mouth. Remember, for Dugesia, it was at the end of a pharynx on the ventral side, whereas for the flukes, if we went back and looked, the flukes, the mouth is there on the anterior end. So it's just a basic comparison that you can answer there. Now, another kind of liver fluke, the cheap liver fluke, belongs to the genus Fasciola, and this one's going to be pretty big here. You can actually see this. If you had to compare it to inches, maybe an inch, 2 inches in length, nothing bigger than that, as this adult form here, or somewhere between 2 centimeters and 5 or 6 centimeters. Not pretty big. You can see it without a scope. So here you can see some of the structures there. There's your mouth area, the oral sucker to help attach in there. Into the body. You can see the word uterus. You associate that with female reproductive structures, testes, where sperm are made. So again, these are hermaphroditic, or another one is monoecious, right? So here, again, sketch, know the genus Fasciola when you see it here, and sketch and label those observations. And then it asks you to go back and look and answer a question about this, and you'd want to go back and look at figure 37.4 and read the caption carefully. It's pretty lengthy, but essentially when you go back and you look at it, it talks about how, and this is that front end of what we were just looking at, It talks about how they can produce up to 3,000 eggs per day throughout their lifetime. And these tons of eggs are released, and this is because for those eggs to make their way into another host would be a very rare event. So the more eggs you can spit out into the environment, the more likely the right host is to pick them up. And so the questions they're asking, well, what is the significance of that when it comes to their evolutionary success? Well, if it's hard to get a host and your lifestyle is that of a parasite, then producing a lot of eggs, so you're an egg maker, right? Then evolution would dictate you have to produce a lot of eggs. A lot of offspring are produced, but not all will survive because of the particular life history of these animals. And then we have the genus Schistosoma, and Schistosoma. They're also flukes, and this one is unlike the others. This one is dioecious. They have separate male and female here, and this one here, the male and the female. The male is larger, and the male's going to have a groove. And when they're mature in the definitive host, say a human, again, or other large mammal, within this definitive host, when the male and female find each other, the female lays inside that groove and they stay together for the rest of their life, and all they're doing is constantly making eggs and sperm and fertilizing the eggs and releasing the eggs. So tons and tons of eggs are produced for the same reason. It's hard to get into the right host overall. So I did find an image of what a microscope slide might look like if you were to look at them in a laboratory under a microscope. And there you can see the larger male there, and then the female within the groove there. And so you want to look at the life cycle of this trematode. And overall, you can see it is an important parasite of humans. And this is the significant thing here. Okay, so the adults live within the human, okay, and they'll live in these are called blood flukes, by the way, because they associate with blood vessels, and they may be associated with blood vessels of the urinary bladder, where they can create a lot of bleeding. You get urine in your blood. So another species may be in another part of or another organ, say the small intestine or so. Depends on the species. So, but they're associated with those blood vessels. So when they're making lots and lots of eggs, the eggs end up getting into the cavity of that organ, say into the urinary bladder or into the intestines, so that the eggs can be released with the feces or with urine. And this can create some bleeding and some other issues. And the fact that there's lots and lots of eggs produced all the time, that causes a great deal of inflammation. So you get some serious health problems associated with this. And I suppose in worst case scenarios, death could occur if you have too big a load of these parasites within you. But as the larvae are going to be released, and here you see some of those names I saw earlier. And the Myricidia, these are ciliated, and they're going to swim. And then they end up getting into a snail-like host here. And then you have your larval Sicariae, which can swim. And in this case, instead of going and penetrating into the body of the skin of a fish, they'll go directly into the human skin. So this is a big problem in tropical areas where a lot of farming takes place within irrigated areas where they use a lot of water, and humans are out there tending to their crops, walking around, and these Cercaria can burrow right into the skin. So you tend to see a lot of these infections with this parasite in areas, especially where this type of agriculture occurs. And again, this can be a big problem because it can cause inflammation of major organs. So all this background here, why is it important? This can affect affects up to 200 million people around the world in temperate regions, tropical regions especially, again, where there's a lot of that kind of farming. And again, these are blood flukes, associated with blood vessels. They can create a lot of bleeding and so on. Now, when you look at this life cycle here, you want to make sure you can identify the intermediate host and the definitive. The definitive in this case is this vertebrate animal, this human here. We have snail, is your intermediate host in this thing. There is no fish in this case. So we know that Schistosoma has separate males and females. They're not hermaphrodites, so we refer to that as dioecious. We saw that term with plants. So how does the shape of Schistosoma differ from that of the flukes we've studied? So you want to check that out and come up with your own response comparing to Fasciola and Opisthorchis. And then we have another class, Cestoda, and these are the tapeworms. And so again, these are in that Neodermata subphylum. This is that other parasitic class. So they're commonly called tapeworms. Okay, and you want to be able to identify the different sections here. Now, something that's pretty interesting about this group is the very highly specialized area where these occur. These are found on the inside of the intestines, where we have digested food already, and they're flat, right? So they don't have a gastrovascular cavity. They don't need one, okay? Because they're already surrounding themselves with digested food from the host. So no gastrovascular cavity like we saw with prior trematodes in the early ones. So they do have an anterior region. It's called the sculex, and the sculex is going to have these suckers and hooks that help them attach. You can see this is the inside wall of the intestine right here, and the head is attached. And then all of these sections, as we move, the youngest proglottids are produced here. These little segments are called proglottids, and they're immature here, and then they end up maturing. And these mature proglottids are going to have both testes and ovaries. So you're hermaphrodite here. And then eventually, as we move towards the back, the eggs have been fertilized, and this is preferably by another tapeworm that's within that intestine as well, because you want to cross-fertilize to promote genetic variation, right? And the mature proglottids eventually will have fertilized eggs, and then we will call that mature proglottid that has fertilized eggs gravid. And gravid is another name for being pregnant, right? Or having fertilized eggs or embryos within there. So gravid you may see associated uses with animals that are carrying fertilized eggs or embryos in there. You'd say that animal's gravid, right? So what structures are found in the proglottids? They're about reproduction. You're going to have ovaries, you're going to have testes, you're going to have a uterus where the fertilized eggs will develop, and so on. So you can see some of the items mentioned here. Now, by the time the proglottids look like this, and they would be at the end over here, they're so mature, the eggs are going to be fertilized, and the uterus would be swollen, filled with fertilized eggs, and we would call that a gravid proglottid. So you're supposed to look at a representative tapeworm called Taenia. And so for Taenia, you're going to look at different sections. And I found an image of an endoscope analysis, and this is a picture of the inside of the intestine using a medical scope technique. You've gone into the intestine, and you can see this tapeworm, and you can see all the proglottids going along some of the length of the intestine there. And this is the beef tapeworm called Taenia saginati. Not saginata. I think that I doesn't go there. But it is in the same genus. There's a pork tapeworm that is a different scientific name. And so you're supposed to study a slide of and so what we see here is a mature proglottid. It's not gravid, so you can see the ovaries right here. You can see the uterus. There's testes in this area here, and some other excretory organs. You got to get rid of waste. This is a genital pore, where we might exchange sperm with another individual here, and then of course release the eggs. So this, you want to be able to, when you look at a slide of this, you're not going to see the sculex, but I did find another image, a photomicrograph, and there you can see different segments of this tapeworm. There's the sculex there, and then there's segments going all the way back. These are immature, and then you have the mature proglottids. So these would be kind of like the one that's in this image from your lab manual, where you have your mature proglottid. And then this picture includes the gravid ones, where the eggs have been fertilized and the uterus is swelled up because it's filled with with the proglottis. Now, the host then, the proglottis at the end with the gravid proglottis, the proglottis will break off and come off in the feces from the host, and that's how the eggs get into the environment. So you want to be able to identify those, and you also want to know this is the genus Taenia. Be able to identify the basic structures of scolex and so on. Then we ask you what is the name of the pork tapeworm, and it's different than Taenia saginata. It's Taenia, and I want you to make sure you look that up. It's in the lab manual. The specific epithet starts with an S, but I'll let you look that up. This another question asked about this is mentioned in the background. So I want you to answer what is significant about this tapeworm. It's a different genus. I'm not having you know this genus name, but I'd just like you to answer this because it's interesting. It's kind of related to us, but I'll let you answer that one on your own. Again, you're responsible for being able to recognize the basic morphology of a tapeworm. Remember, they don't have a gastrovascular cavity, and they're made of a head, neck, and these proglottids. Be able to identify those basic structures that are listed here. We're not going to go as far as me asking you to identify ovaries or uterus. Some instructions might make you do that. I just want you to be able to recognize a tapeworm and know these representative names for them. So this is the life cycle you see on the right for Taenia, and you can see a micrograph of the scolex and so on. And this is the pork tapeworm. It is the genus Taenia, but I'm asking on that prior question, what is the entire full scientific name for it? You can see your mature proglottid broken off in the feces, and then it gets picked up by an animal. And then here, the pork tapeworm, the larva gets encysted within the flesh, the muscle of the animal, and then, of course, we eat pork chops and bacon and all that kind of stuff. And if it's not cooked very well, and you don't kill the larva, you could get infected with this tapeworm. So make sure you go through this and answer these questions about question 5. It's several sub-questions about this. And as usual, you always go back and you look at the first objective and see what its fundamental process is overall. The letter B asks about how tapeworms obtain their food if they don't have a gastrovascular cavity. I just talked about where they live. They live in a soup of digested food and so on. So make sure you answer those for your own, so you think about what you're looking at a little more closely.